Hello again, everyone. This is the fourth and final lecture for Chapter 2. This is also the final lecture for Unit 1, and everything up to this point is fair game for our first lecture exam. Everything that comes after this will not be on the first lecture exam. So today what we're going to be looking at is some more biochemistry, but today we're going to be looking at the organic biochemistry. And as always, let's start with our not really attendance questions. First, a solution with pH 4 has blank times blank H plus than a solution with pH 6. Well, in this case, the first blank is going to be a number, and the second blank is going to be either more or less. Next, you tell your doctor that you are experiencing pain in your heel. What regional term applies to this area? So go ahead and try to answer those on your own. Pause the video before listening to see what the correct answers are. And let's see. A solution with pH 6 or pH 4 has how much more or less H plus than a solution with pH 6? Well, first, if pH 4 compared to pH 6, first thing we have to do is look to see, does pH 4 have more or less H plus than pH 6. Remember, the lower the number, the greater the H plus concentration. So pH 4 is going to have more H plus concentration than pH 6. How much? Well, remember, every time we go down one whole number, it's a tenfold increase. So 5 has 10 times the H plus concentration as 6. 5 has 10 times the H plus concentration as 6. 4 has 10 times the H plus concentration as 5. So what we do is we take 10 times 10, since we went down 2 steps. pH 4 has 100 times more H plus concentration than a solution of pH 6. Next, you tell your doctor that you are experiencing pain in your heel. What regional term applies to this area? And here we have some choices. Well, cephalic means head, so it's not that. Tarsal is ankle, so we're getting close, but that's not it. Coxal is hip. Olecranal or olecranal is elbow. Calcaneal is the heel. So when you tell the doctor that you have pain in your heel, he's going to say that there is pain in your calcaneal region. Now, as I just said, this lecture is going to be focused on organic biochemistry. We covered the inorganic side previously, and we kind of mentioned already that the difference was organic compounds contain carbon, or inorganic compounds may or may not contain carbon. Most inorganic compounds do not contain carbon. Now, there are some exceptions. Carbon dioxide, CO2, a carbon monoxide, CO, they both contain carbon, but they are considered inorganic. So it can be a little bit tricky, and I'm not just outright going to ask you, is this organic or inorganic? But let's take a little bit deeper look as to why that is, just because I know it can be confusing. Even though carbon is what makes something organic, typically when we're looking at organic compounds, they also contain hydrogen and oxygen. Now, carbon can bond with up to 4 atoms. We saw that back in our lecture on elements and how they interact with each other. Remember, carbon forms covalent bonds. It does not give away, it does not take electrons. It shares electrons and it shares them equally, so it forms covalent bonds. Now, remember, carbon can form 4 single covalent bonds, but it's also capable of forming double covalent bonds or even triple covalent bonds. And in organic chemistry, we're going to be looking at some really big molecules. And we'll see the biggest molecules in the body later in this lecture. But when we talk about these big molecules, we give them names. We call them macromolecules. And macro is just a root word that means large. So macromolecules are just really big molecules. And these molecules tend to be made of small units that repeat over and over and over. Now, when we have that kind of layout, when we have a big structure that's actually made of repeating smaller structures, we call the big structure a polymer. A polymer is just a big structure made of smaller pieces. And those smaller repeating pieces we call monomers. So the molecules that we're going to be looking at today, most of them are polymers with repeating monomers that make them up. And we're going to give examples of these as we go on, but I want to go ahead and give kind of a way of thinking about polymer versus monomer to get kind of get it locked in your head. Think about a brick house. Well, the house is made of small bricks over and over and over again. The house would be the polymer in this case. And the repeating pieces, the bricks, those would be the monomers. A brick house is a polymer. The bricks that it's made from are the monomers. And the molecules that we're going to look at today, most of them are going to follow that scheme. And there are 4 classes of organic compounds. There are carbohydrates, there are lipids, there are proteins, and there are nucleic acids. Carbohydrates, lipids, proteins, and nucleic acids. And we're going to look at all 4 of those in detail to see what do they look like, what is their function, what are they made of. So right now, all we've done is just introduce those 4. But now we're going to go through and look at each of those 4 in more detail. And the first class that we're going to look at are the carbohydrates, which are sugars and starches. And when we talk about carbohydrates, the things that they are going to be made from, the elements that make up carbohydrates, are going to be C, carbon, H, hydrogen, and O, oxygen. Those are the only things that will ever make up a carbohydrate. Carbon, hydrogen, and oxygen. And almost always, that carbon, hydrogen, and oxygen are going to be in the ratio of 1 to 2 to 1. Sometimes you will even see it written as CH2O. Now that doesn't mean there is only 1 carbon, 2 hydrogens, and 1 oxygen. It means that there is always going to be an equal amount of carbon and oxygen, and there's going to be twice as much hydrogen. There can be small variations on this, but almost without fail, carbohydrates are going to have carbon, twice as much hydrogen, and then oxygen in the same amount as we saw with carbon. For example, in the previous lecture, or maybe the lecture before that, we looked at the equation for cellular respiration, and we talked about glucose. Well, glucose is a carbohydrate, and the symbol for or the equation for glucose was C6H12O6. C6H12O6. Well, if we look at that, C6 and O6, well, those are equal. And H12, well, there's twice as much hydrogen as there was carbon and oxygen. So that's how the carbohydrates are structured. And usually when they are drawn in a textbook, they're drawn like we see over here on the right. Now, first let me just say, I know that when a student sees something like what we have here in this box, immediately their brain starts to shut down because it can look overwhelming. I want to tell you right now, don't worry about that. This is just so that I can show you some basic structure of carbohydrates. You will never have to read one of these. You will never have to produce one of these. If I show you one, you will not have to name it. This is just so I can explain carbohydrates visually. Now, normally when we draw carbohydrates, we're going to see ring structures like we see here and down here. So these rings represent kind of the shape of the molecule. And when we're talking about carbohydrates, they are going to follow that polymer-monomer structure that we talked about. The monomers for carbohydrates are called monosaccharides, or simple sugars. Monosaccharides, and that's what we see across the top row here. Now, when we draw them, like I said, we draw these ring structures. And in monosaccharides, which are, again, the monomers for carbohydrates, we have a single ring all by itself. And if we look at these letters, we see H's, we see O's, and we see C's, nothing else. Now, there are some other letters there that we don't see because chemists like to do this weird thing to where when there's a bunch of lines that come together, there's a C there, and they don't show it. If there's something other than a C there, then they do show it, like we see right here with oxygen. But again, don't worry about that. Just look to see what letters do you see. We see H, O, and C. That's all that we should see when we're looking at carbohydrates. And monosaccharides are a single ring all by themselves made of carbon, hydrogen, and oxygen. Here the same thing, a single ring made of carbon, hydrogen, and oxygen. Now, here we see some examples, and you won't have to know these examples, but some of them you're going to get used to because we talk about them a lot. We have glucose. Glucose we're going to talk about a lot this semester. We've already spoken about it. Fructose. This is fruit sugar. Over here we see deoxyribose and ribose. We're going to talk about those a few times in this lecture. And when we talk about these monosaccharides, we're going to talk about how much carbon is there. And the number of carbons is pretty much always going to be equal to the number of sides of this ring. So if we look at this glucose we said C6H12O6, 6 carbons, we have 6 sides. Fructose is a 5-carbon sugar. We have 5 sides. Deoxyribose and ribose, they have 5 sides. Well, now there is some catch to that. This fructose, even though it has 5 sides, technically it has 6 carbons because there's a carbon kind of hidden away right here and a carbon hidden away right here. So it doesn't always follow that scheme, but as a general rule, the number of carbons is equal to the number of sides. Okay, so how do we name these sugars? Well, we have the name of the individual sugars, which we see right here, but we can also kind of classify them by how many carbons does it have. If it has 6 carbons, we call it a hexose. If it has 5 carbons, we call it a pentose. And we can build upwards from that. So we can see that just because something is C6H12O6 doesn't necessarily make it glucose. It could also be fructose, it could also be galactose, it could be a few others. Same thing with deoxyribose, ribose, and a few others. Now the monosaccharides, single rings, are the monomers, but we can start building things. We can start joining them together. If we have 2 monosaccharides and we join them together, now we have 2 rings with a bond formed between them. These are called disaccharides. Disaccharides are 2 monosaccharides joined together. And some common ones, Here we have sucrose. Sucrose is a glucose and a fructose joined together. Sucrose you probably all have in your kitchen right now. This is table sugar. Over here on the right, we have a galactose and a glucose joined together. This makes something called lactose. This is the sugar that's found in milk. And incidentally, if you've ever heard someone say they are lactose intolerant, what that means is they are missing the enzyme that breaks this bond. They can't break lactose down. We could keep joining these together over and over and over, and we get these long chains. These are called polysaccharides. Poly for many. It's a lot of monosaccharides joined together. And a couple of common polysaccharides are, well, starch, which is a plant molecule. Plants store energy in the form of starch. Humans do the same thing. We store extra glucose in long chains, except when we do it, we form something called glycogen. Glycogen is how humans will store excess glucose. We do that in the liver and a few other places around the body. So what are carbohydrates for? Well, they are very, very easy to get to sources of quick energy. When our body needs energy very quickly, we break down carbohydrates and release energy to power our body. Now, sometimes, and we'll see this when we get to chapters 3 and 4, talk about the cell, carbohydrates can also form structural components at the cellular level. Cells will hold on to their neighbors using carbohydrate chains. Certain parts of our body's architecture, the framework that builds our tissues, can be carbohydrates. Now, these polysaccharides, like I said, they're stored away and they are good sources of quick energy, but they're not very soluble in water. So if you put starch in water, it's not going to dissolve very easily. Eventually it will, but it's not going to dissolve very easily. So carbohydrates, primarily quick sources of energy that's very easy to get to. Secondarily, they are structural. Now, the next class that we're going to look at are lipids. Lipids are fats and oils. And these are also not very soluble in water. So if you think about, if you've taken a drop of oil and put it in a glass of water, what happens? It doesn't mix. So lipids are not soluble in water. And a term for that, or actually a couple of terms we're going to introduce here, they are hydrophobic. Hydro means water. Phobic or phobia means fearing. Lipids are hydrophobic. They are water-fearing. They do not mix with water. A term that means the same thing is non-polar. Lipids are hydrophobic. They are non-polar. Think back when we introduced the word polar. Well, polar, another word for polar, is hydrophilic, water-loving. So things that are polar are hydrophilic. Things that are non-polar are hydrophobic, and lipids are hydrophobic. When we talk about lipids, fats and oils, they do not follow that scheme. They do not have monomers. So they don't follow the polymer monomer scheme that all the others that we're going to look at today follow. Lipids are very big, and they are made of smaller pieces, but they're not repeating smaller pieces. So they do not have monomers. And there's a few different types of lipids. There are triglycerides, phospholipids, steroids, eicosanoids, and we're going to look at these, although the eicosanoids we're not really going to talk about all that much because you don't need to know about them. I'm not going to ask you about them on exams. I will tell you what they are when we get to that point, but you will not need to know that for exams. So first, let's look at the triglyceride lipids. Triglycerides, when they are solid, we call solid at room temperature, we call them fats. When they are liquid at room temperature, we call them oils. Now, that's just kind of, fats and oils is not a clear-cut division between the two because some oils are solid at room temperature, some fats are liquid at room temperature. But as a general rule, fat just means solid at room temperature and oil means liquid at room temperature. And when we're talking about triglycerides, they have a common structure. First, we have these long chains of carbon surrounded by hydrogen. And these are called fatty acids. Here we have a fatty acid, here we have a fatty acid, here we have a fatty acid. And the three fatty acids are going to be joined to something called glycerol. We take three fatty acids and join them to a glycerol. So now we have a molecule that looks like this, a glycerol molecule with three fatty acids coming off of it. That is a triglyceride. And its functions are energy storage. Well, really, fat has a lot more energy than the carbohydrates that we looked at previously. The difference is carbohydrates are really easy to break down and get the energy out of. Fats are really difficult to break down. Even though they have more energy, it's harder for our body to get to it. So this is more of a long-term energy storage molecule. Also, insulation. Fat is really good at holding in heat. So we have a layer of fat just beneath our skin, and it helps insulate our body. And protection. Fat, in addition to being an insulator, it's also a cushion. Like I just said, we have a layer of fat just beneath our skin, so if we fall, it helps as a shock absorber. Also around certain organs, like around our kidneys, behind our eyeballs, there's fat there, and it acts as a cushion or a shock absorber to protect us. Now, I want to talk about something called saturation. We've all heard of saturated fats, but we might not know exactly what that means. So up here at the top, above the red line, I've got a triglyceride, and down below the red line, I've got a different triglyceride. Up at the top, this is a saturated fat, and down at the bottom, this is an unsaturated fat. So let's see what the difference in the two is. Well, when we're talking about a fat that is saturated, we're going to look at the fatty acids. Look at these long chains of carbon. Now, if we look at these long chains of carbon, we can see every carbon here has how many covalent bonds? Every single one of them has 4 covalent bonds. And every single one is covalently bonded to either a carbon or a hydrogen at each of those bond sites. This carbon has 2 hydrogens and 2 carbons. This carbon has 2 hydrogens and 2 carbons. When we get to the end, this carbon has a carbon and 3 hydrogens. So every single one, not only does it have 4 covalent bonds, those bonds are all single, and there is as much hydrogen here as we could possibly hold. So saturation in general just means something is holding as much as it can. When we're talking about saturated fats, it's talking about the number of bonds and the number of hydrogens that is attached to the molecule. There is the maximum amount of hydrogen. There are the maximum amount of covalent bonds because they are all single bonds. And if we look, we could see that these chains are very straight. and that gives it its firmness. That makes it a solid at room temperature. Let's compare that to an unsaturated fat, which we see down here at the bottom. Now, here we see again the same thing that we saw above. And the second row, again, same thing that we saw above. But if we look at this bottom fatty acid, this carbon, instead of having 4 single covalent bonds, it actually has 2 single and a double covalent bond between itself and the next carbon. Well, each of those carbons now only has 3 bonds. It has 2 singles and a double. So technically it's 4 covalent bonds, but one of them is a double, so we'll just count it as 1. The result is each of these carbons could potentially hold another hydrogen there. It could form another single bond if we broke this double bond. So we do not have the maximum amount of hydrogen. There's less hydrogen here than there is up here in this top triglyceride. The more double bond carbon-carbon we have, the less hydrogen we will have. And if we look at what it does, it puts a kink in the chain. When there is a double bond, it makes the chain bend. It puts a kink in the chain. The more double bonds there are, the more kinked up the chain will be. These tend to be liquid at room temperature, oils, and they are more heart healthy. Why is that? Well, in order to break these fats or oils down, water has to be able to get to the bonds. It has to be able to get in there to actually break those bonds with a hydrolysis reaction. Up here at the top in the saturated, well, since the chains are so straight and packed tightly together, it's difficult for water to get in between those chains. But down here, where there is a kink, water can get in here much more easily to start working on those bonds to break them apart with hydrolysis. So unsaturated fats and oils are easier to break down, which makes them heart healthy. The more unsaturated they are, maybe you've heard the term polyunsaturated, that just means there are a lot of these double bonds. It makes it much easier to break down. 2 other terms that you've probably heard are trans fats and omega-3 fats. So I just want to talk about what they are real quick, but we're not going to spend a lot of time on those. These are both triglycerides. Trans fats are oils that have been modified so that they are solid at room temperature. Even though they have some double bonds, they still come across with these very straight chains as opposed to these bent chains. They've been modified so that their chains are straight, which makes them solid at room temperature, which makes them very unhealthy, makes them harder to break down. Omega-3 fatty acids are very heart healthy. These are the fats that we associate with certain fish. Fish has a lot of omega-3 fatty acids, which are very good for our heart. They are fats that are easy to break down. Okay, moving away from triglycerides, the next thing we're going to talk about are phospholipids. And we see a phospholipid over here. Now we see this depicted 3 different ways. Here on the left, this is kind of what we just saw previously with our carbohydrates and our triglycerides. This is kind of the chemical way of writing it. Here in the middle, this is more of the molecular drawing. But over here on the right, this is how we're going to see this same molecule quite a bit this semester. It looks kind of like a tadpole with 2 tails. So this is a phospholipid. Let's look at what a phospholipid actually is. Phospholipids begin as triglycerides, but one of the tails is removed so that it's only got 2. And on the other side of the glycerol, we add a phosphate group. Now, the phosphate group will have some other stuff going on with it also, but it's a phosphorus-containing head group. So over here, these two tails, those are the fatty acids. And then the head is the phosphate group. These are very important because it sets up a neat situation, whereas the tails, since they are fatty acids, they are non-polar. They are hydrophobic. But the head is actually polar. It likes water. It's hydrophilic. So we have this one molecule where one side of it is non-polar and the other side of it is polar, which makes actually the whole molecule polar. Because remember, the word polar, like I said, it's going to keep coming back all semester long. The word polar just means one area of something has different properties than another area. In this case, the head has polar properties, the tail has non-polar properties, so the entire thing is also a polar molecule. This is going to be very important when we start talking about the cell membrane in chapters 3 and 4. The cell membrane is primarily made of these phospholipids. So we're going to see this kind of tadpole-looking picture quite a bit over the next couple of chapters. Here we are going to talk about cholesterol. And what we see in this image is kind of the basis for a steroid hormone. Now, cholesterol gets kind of a bad rap in that we think about it and we think, oh, cholesterol, it clogs your arteries, it's bad for your heart, which is true if there's too much of it or if there's certain types of it. But cholesterol is actually really important for our body because it does things like it also forms part of our cell membranes. It forms the basis for all of our steroid hormones like testosterone, estrogen, aldosterone, cortisol, lots of others. And that's because we start with a molecule of cholesterol and add a few other things to it. And depending on what other things we add to it, that gives us different steroid hormones. Also, vitamin D is based on cholesterol. So cholesterol actually has a lot of very important factors for our body. And finally, eicosanoids. Remember, I said eicosanoids, we are not going to have any information about this on the tests. I just want to briefly mention them because your book does. Eicosanoids are lipids that are involved in things like blood clotting, blood pressure maintenance, inflammation processes, which are an immune response that our body performs, labor contractions during delivery. But again, don't worry about eicosanoids. They will not be on exams. Okay, so now we're going to talk about proteins, our third class of macromolecule. Proteins are going to be made of carbon, hydrogen, oxygen, and nitrogen, and some of them will also include sulfur and phosphorus, but all of them will have C, H, O, and N. Now, protein is a macromolecule that is a polymer, and it does follow the polymer-monomer scheme. Its monomers are small structures called amino acids. We see a couple of examples of amino acids here. We see methionine, cysteine, tyrosine, and arginine. And there's actually 20 different amino acids. You do not need to know any of them, although it is a good idea to know their general structure. So what we do with amino acids to make a protein is we will take one amino acid and another amino acid. We perform a dehydration synthesis reaction that causes a covalent bond to join between the two amino acids. And even though it's a covalent bond, we give it a special name. We call it a peptide bond. A peptide bond is just a covalent bond joining two amino acids together. Now, if we go back and look at these amino acids, if we look at the black area, ignore the pink, if we just look at the black area here, it's exactly the same. If we had all 20 amino acids shown, the black area outside of the pink box, the black area would be exactly the same in all 20 of them. The part that is different is this pink box area, and it's different for all 20 amino acids. This is called the reactive group, or the R group. And the reactive group can look a lot of different ways. It could just be a hydrogen. It could be a long chain. It could be a short chain. It could be a chain with a ring structure in it. It could be a branching or charged chain. Some of them will have hydrophilic properties. Some of them will have hydrophobic properties. Some of them will be acidic. Some of them will be alkaline. They're all different, but it's only that reactive group that is different. If we go back and just look at the part that is similar, there's 3 different regions to it. There's a carbon right in the middle, the central carbon, that has a hydrogen on one side of it. It has a nitrogen with 2 hydrogens on one side of it. This is called the amine group. And it has a carbon double bonded to an oxygen, single bonded to an OH group. This is called the carboxyl, or the carboxylic acid group. So carboxylic acid and amine, that's where we get amino acid. Now it is the R group that is different. Here we just see an R. This does not mean that R is a chemical symbol for something. There is no chemical symbol R. This just means that reactive group. And this is what's different for all of them. So it is the R group that gives the amino acid its properties. When we go to build a protein, we're going to take those amino acids and join them together, one after another, after another, after another. That's what we see up here at the top. Each one of these little colored balls represents an amino acid. And the order of amino acids is called the primary structure. Primary structure is just a chain of amino acids. Whatever order they're in is the primary structure. Now remember, all of these amino acids will have R groups coming off of them. And the R groups are different, and they have different properties. And based on their properties, it's going to cause this chain to start to fold up and kink up and coil up. And sometimes it forms this kind of corkscrew pattern called an alpha helix. Sometimes it folds back and forth like corrugated cardboard. This is called a beta pleated sheet. But based on those R groups, we get different regions that have these different structures. This is called the secondary structure. But the secondary structure is based on the primary structure, the order of amino acids. And once we have these secondary structures in place, they are going to start to interact with each other. And the whole thing starts to fold up and take on a 3-dimensional shape called the tertiary structure. And at this point, we no longer just have a chain of amino acids, we have a protein. Once we get to the tertiary structure, we have a protein. But where did that tertiary structure come from? It came from the secondary structures. And where did the secondary structures come from? It came from the primary structure, the order of amino acids. If you change the order of amino acids, ultimately you change the shape, the tertiary structure. So proteins, it's all going to be about shape. Shape is essential for proteins. That's what determines what job they have, how well they do their job, how they do their job. It's all about shape. And if you add anything to this shape. Or if you take anything away from this shape, it's going to change its shape, and it's going to change its properties. Proteins are all about shape, and its shape is what allows it to have the properties to do its job. That is going to come back all of Bio 137 and all of Bio 139. Proteins are all about shape. Now, we stopped here at tertiary structure because that's when it becomes a protein. Sometimes we will have more than one protein stick to each other, and we get something called quaternary structure, which is what we see down here. This is actually a protein called hemoglobin. Hemoglobin is made of 4 proteins stuck together. It could just be 2 proteins sticking together, but whatever it is, we have multiple proteins sticking together to get something called the quaternary structure. Now, once we get to the tertiary level, like I said, that's its shape, that's what gives it its properties. And we can put these kind of in 2 broad categories. Sometimes the shape is going to be very long and thread-like. We call these fibrous proteins. They tend to be structural. They're going to do things like give structure or give shape to things in your body. Things like keratin, which is what's in your skin to give it its firmness. Things like elastin and collagen, which are in our connective tissues. Or we could have something that looks more like what we see down here at the bottom. We could have a globular protein. Globular proteins tend to be functional. They tend to do things in our body. For example, a lot of hormones are globular proteins. Antibodies, which are molecules that are helping to keep us from getting sick in our immune system, those are globular proteins. Hemoglobin is a globular protein. But again, it's all about shape. And if you change a protein's shape, you change its ability to do its job. So what happens if we break some bonds in this protein? Well, if you break its bonds, you're going to change its shape. And that's called denaturation. Denaturation is what happens when you break bonds within a protein and cause it to lose its shape. And the 2 most common ways of denaturing a protein are with increased temperature and decreased pH, by making it hotter or by making it more acidic. High heat, low pH will break bonds and denature a protein. For example, if you've ever fried an egg, when you crack the egg, that white part is very liquid. It doesn't have any real color to it. It's a protein. If you put it in a hot skillet, what happens? Well, 1, it changes color, so it looks different. 2, it starts to firm up. It feels different. We've changed its properties by increasing the temperature. That's denaturation. Enzymes. Now, we've talked a little bit about enzymes already, back when we were talking about catalysts. Well, enzymes are biological catalysts, and they are proteins. Typically, they are globular proteins. Now, remember, enzymes, catalysts in general, what they do is they will speed up the rate of a reaction. The way that books will say it is they will lower the activation energy. Don't worry about that. That's more of a chemistry way of saying it. We will just say that it speeds up the rate of reaction. And it can actually speed it up so that something that would not happen in your body within your lifetime, because it happens so slow, it speeds it up to millions of times per second. And when we look at enzymes, again, their shape is what allows them to do their job. They are very specific. This enzyme that we see in the image here, its job, I guess, is to take this structure here and break it in half. Well, the way that enzymes work is they will have a pocket so that whatever job they're going to do, whatever they're going to work on, that substance will fit into that pocket. That pocket is called the active site. And whatever fits into the active site is called the substrate. So the substrate here is going to fit into the active site of the enzyme, and the enzyme is going to break it apart. So let's see how shape works. The substrate is shaped so that it fits perfectly in the active site. This enzyme, his active site is shaped in such a way that its substrate will fit into it perfectly, but other things will not fit. So his job is very, very specific. Well, the substrate comes along, it fits into the active site. What have we done? We've added something to this protein. And what did we just say happens when we add something to the protein? It changes shape. So these arms here kind of bend inward, it looks like. And when they bend inward, it's going to apply some pressure to this point right here and break it apart. Well, once we break this apart, the substrate is actually shaped a little bit different. It doesn't perfectly fit into the active site anymore. So the enzyme doesn't like the substrate anymore. It kicks those two products out. Now what have we done? We have removed something from the protein. So what happens? It goes from this shape back to its original shape, which is important because remember when we talked about catalysts before, one of the things we said was not only do they speed a reaction up, they are unchanged by the reaction. So after this reaction happened, the enzyme is exactly like it was before the reaction happened. So enzymes, they are proteins, they are biological catalysts. They are very, very specific based on their shape. And how do we name them? Well, anytime we look at an enzyme, chances are it's going to end in the letters ASE. ASE. For whatever reason, enzymes tend to end in ACE. And the first half of the word usually tells us something about what job the protein does. So for example, when we look at the cell membrane in the next chapter, we're going to see that it has two layers, an outer layer and an inner layer. And sometimes there's something on the outer layer that we need to move or flip to the inner layer. And there's an enzyme that takes that small section of the membrane and flips it. So, scientists, I guess trying to be clever, when they discovered that enzyme, they gave it the brilliant name of flipase. ACE because it's an enzyme, and it's an enzyme that flips a part of the cell membrane. Flipase. All right, so now let's talk about the last of our 4 classes of organic macromolecules. Now, most of the stuff that we're going to talk about now is actually going to come up in chapter 4. I like to introduce it here because it ties in with the organic macromolecules, and that way when we get to chapter 4, it's more of a review than a whole bunch of new material. Now, when we talk about nucleic acids, primarily what we're going to talk about are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. There are others, and we'll mention them, but those are the main 2 that we will talk about. They are the largest molecules in the body. And actually, DNA is surprising how large it is. DNA is in every cell in your body. It's packed into the nucleus of every cell. And DNA, when you unwind it, is over 6 feet long. Well, you have about 100 trillion cells in your body. Every one of them has about 6 feet of DNA in it. So imagine how much DNA is in your body. All right, well, DNA and RNA, they are nucleic acids. Nucleic acids, they do fit our scheme that we've been following. They are polymers. And the monomers that they are made of are called nucleotides. Nucleic acids are the polymer, nucleotides are the monomers. And what we see over here is a nucleotide. This is just a generic nucleotide. Now, if we look at DNA, DNA is always going to be made of carbon, oxygen, hydrogen, nitrogen, and phosphorus. So that's all that we will ever see in our nucleotides. Now, all nucleotides follow the same structure. Here is just a generic nucleotide, so let's see what structure it has. Right in the middle, it's going to have a 5-carbon sugar, either deoxyribose if it's DNA, or ribose if it's RNA. Either way, it's a 5-carbon sugar. On one side, it's going to have a phosphate group. On the other side, it's going to have something called a nitrogenous base. Now, the nitrogenous base, if we just look at it quickly, it looks like it might be a sugar because it's got this ring-shaped structure. But it's not, and the way that we can tell is remember what carbohydrates were made of. CHO, and that was it. Well, we see this one has a nitrogen in it, so it is automatically not a carbohydrate. It's not a sugar. This is a nitrogenous base. All nucleotides have a 5-carbon sugar, a phosphate group, and a nitrogenous base. So let's look at DNA. DNA is what we see right here. And if we look at the nucleotides, here's a couple of examples. Here we have one nucleotide. Here's our 5-carbon sugar, deoxyribose. On one side, we have a phosphate group. On the other side, we have our nitrogenous base. Over here, we have the same thing. Here's our 5-carbon sugar, deoxyribose. On one side, we have our phosphate group. And on the other side, we have our nitrogenous base. Now, when we're talking about DNA, it's the nitrogenous base that determines which of our nucleotides we have. Here we have one nucleotide called adenine. Over here we have a different nucleotide called thymine. And there's 2 others, cytosine and guanine. So how do you know which one you're looking at? Well, first we have to go back and look at the structure of our nitrogenous base. Sometimes the nitrogenous base will have 2 rings. Sometimes the nitrogenous base will only have 1 ring. If there are 2 rings, we call it a purine. Purines have 2-ring nitrogenous bases. One of them is adenine, which we just abbreviate A. The other is guanine, which we abbreviate G. So the purines have 2-ring nitrogenous bases. They are either A or G. If they only have 1 ring, like we see over here with the yellow one, it's called a pyrimidine. Pyrimidines are thymine, abbreviated with a T, or cytosine, abbreviated with a C. So our 4 bases for DNA are A, G, T, and C. Now DNA, which we see kind of drawn out here in the classic cartoonized way that we see DNA all the time, it's called a double helix. A double helix is like a ladder that somebody took and twisted. And if we see it here, we can see that the ladder has the legs that are twisted around, and the rungs across the middle hold those 2 legs of the ladder together. Well, the rungs of that ladder are those nitrogenous bases. The legs of the ladder are where we find the deoxyribose and the phosphate. So right in the middle, the rungs holding the 2 sides of the ladder together are these nitrogenous bases. And the really cool thing about it is the nitrogenous bases, they're always going to be in a very, very specific layout. They could be in any order. Every single one of us has a different order, but they're always going to follow the same scheme. And this is called base pairing. If we look at the nitrogenous base on one side, if we look and see that it is an adenine, an A, it is always going to have a thiamine, T, on the other side. Adenine and thiamine are always joined together. If on one side we see that it is a cytosine, it is always going to be a G, a guanine, on the other side. Cytosine and guanine are always joined together. That's the base pair rule. A and T go together, C and G go together. So if someone tells me, I found some DNA, and on one side the sequence is A, C, A, G, C, well, you automatically know what the other side of the DNA is, even if you don't have it. The other side is going to be T, G, T, T, C. You can figure it out if you know one side because of base pairing. So where do we find DNA? DNA is found inside the nucleus of our cells. And what does it do? Well, it contains our genes. But that's not a good enough definition because we haven't really talked about what genes are yet. So what does DNA do? Well, it provides instruction for protein synthesis. DNA tells our cells which proteins to make and when to make them. And that'll be the topic of Chapter 4. Chapter 4 is going to look at how that happens. So keep that information handy because we're going to come back to it and build on it. Okay, changing gears just a little bit, still talking about nucleic acids, but now we're going to talk about RNA instead of DNA. RNA is ribonucleic acid, and it's very similar to DNA in structure. It's only a single strand instead of a double strand. It's still one long fiber, but it doesn't have a second side to it. So it's not like a ladder. It's like somebody took a ladder and sawed it in half. And the 5-carbon sugar is ribose instead of deoxyribose. Now the bases are very, very similar. We still have adenine. We still have thymine. We still have adenine. We still have, let me go ahead and write this down on our whiteboard so I don't confuse you more. Here's our RNA. It's just one-sided. We still have adenine. We still have cytosine. We still have guanine. But there is no T, there is no thymine in RNA. Instead, there is a different nucleotide called uracil, abbreviated U. So RNA still has adenine, still has cytosine, still has guanine, but instead of thymine, it has uracil. And it does not have the second side. It is only single-stranded. And there are a lot of different types of RNA, way more types than we will cover in our class. But there are things called messenger RNA, abbreviated mRNA. There's transfer RNA, abbreviated tRNA. Ribosomal RNA, or rRNA. And lots of others. Now, when we get to chapters 3 and 4, we're going to talk about what all of those types do. But again, most types of RNA we will never talk about in this class because there are just way too many. Now, finally, adenosine triphosphate. This is also a nucleic acid. And ATP, or adenosine triphosphate, which is what we see here, this is actually going to be a molecule that we talk about in almost every single chapter in both 137 and 139. ATP is like the gasoline that our cells run on. Every time there's a reaction that requires energy to proceed, ATP is probably what's powering that reaction. So let's look to see what ATP actually is. Here we have a 5-carbon sugar, ribose. So it sounds like it's going to be RNA related, and it is. On one side, we have a 2-ring nucleotide, or a 2-ring nitrogenous base, adenine. Well, ribose plus adenine is called adenosine. If we add a phosphate group onto that, it's called adenosine monophosphate, or AMP. If we add a second phosphate group onto it, it's called adenosine diphosphate, or ADP. And if we add a third phosphate group onto that, it's called adenosine triphosphate, ATP. Now, there's a lot of energy stored in these two chemical bonds here. Those phosphate groups, they kind of don't really want to be attached to each other. So it's almost like if you take a spring and you push your thumb down on it, there's a lot of energy stored in that spring. As soon as you take your thumb away, that energy is released and the spring bounces. Well, if I could get rid of one of these phosphate groups, then I would release that energy, and that energy could power a chemical reaction. So let's see how ATP works to power chemical reactions. If a molecule of ATP comes along and gives away one of these phosphate groups to another molecule, we say that we have phosphorylated the other molecule. Phosphorylation is taking a phosphate group from one molecule and putting it onto another molecule. And when I do that, when I transfer this phosphate group to another molecule, this bond is broken and energy is released. And that energy can then power a chemical reaction. But once I do that, once I've taken this phosphate group off, once I've phosphorylated something and released that energy, what am I left with? I'm left with a DP, adenosine diphosphate. And there's still a little bit of energy left in this bond right here. And every now and then, in certain circumstances, a DP can phosphorylate someone, releasing a little bit more energy to power another reaction, leaving us with a MP, adenosine monophosphate. That's a lot less common. It is almost always a TP that we see doing that kind of activity. Okay, so we are now done with chapter 2. We're done with chemistry. The good news is there's no more new chemistry in all of Bio 137 or Bio 139. We're done with it. We will not learn any more new chemistry. The bad news is you really do have to understand the chemistry that we've covered in chapter 2. If you didn't, please make sure to ask me questions. Make sure to ask a tutor questions. Make sure to look at things on Khan Academy or YouTube or whatever it takes to get this to click, because we are going to use the chemistry that we've learned. And you do have to understand it, because once you get through chapter 2, it's assumed that you understand it. And when we talk about something like a redox reaction, well, you already know what a redox reaction is, so I'm not going to explain it a second time. So make sure that before we move on, any questions that you have, you either ask me or get it cleared up somehow. All right, well, that is the end of chapter 2. That is the end of unit 1. And next up is our exam. And everything up to this point is fair game on the exam. Everything that comes after this will not be on exam 1. Okay, take care and let me know if you have any questions, and I will talk to you next time.